So the next step indeed is the detection. So how does this work? Well, you shine a laser through your flow cell. And that laser will activate the labels on the DNTPs that have been incorporated. So these DNTPs, these labels, will start emitting light. And then the camera comes in and the camera will make pictures of every tile on the flow cell. So hundreds of pictures. And this is what it typically looks like. When you prepared the flow cell for sequencing, you had no control whatsoever where the clusters will form. Yeah? You don't know where they are. So in the first cycle of sequencing, the first thing that you always have to do is you have to see where the clusters are. How do you think this can be achieved? Ooh. So at this point, the only thing you have is a set of pictures of your flow cell. And on this set of pictures, you need to know where clusters are located. So basically, what the software, the Illumina software, will look for is regions on the flow cell where he can see a signal. And he's not going to interpret that signal, so he's not going to see which color it is. He's just going to see where is a signal above background. So in all the spots that he can find, where he can see a detectable signal, are considered clusters. And then, in the next step, he is going to interpret the signals. So he's going to see which color they have, which wavelength was emitted. And of course, since every cluster contains identical copies of one fragment of your library, the whole cluster will emit the same light. So here, for instance, you have a cluster emitting red light, so he knows, red is a T, so in that cluster a T has been incorporated. And the same is true for this one. And in this cluster he can see, the whole cluster is emitting yellow light, yellow is A, so in that cluster an A has been incorporated. And this is called base calling. So he's interpreting the color of each cluster and he is linking that color to a certain base. And then you can proceed and you can do the next sequencing cycle. So in the next sequencing cycle, you're going to cleave off. The first thing that you do here is you're going to cleave off the label, the color, and you're going to cleave off the terminator. Otherwise, you cannot do a second cycle. And you're going to repeat the process again. So again, you're going to build in one single base in every cluster on the flow cell. You're going to shine a laser through the flow cell and the camera is going to take pictures and the software is going to call the second base for every cluster. Now here at this point, the software already knows where the clusters are. So he knows, in the first cycle of sequencing, I found that there's a cluster here in the corner. So in the second cycle of sequencing, that cluster is still there, and now it's emitting blue light. Blue is a G, so now a G has been incorporated there. And the same thing is true for this one. So in the first cycle, this one was emitting an A. In the second cycle, it's green, so now it's emitting a C. And you repeat this process for many, many cycles. So again, typically for around 100 cycles. And in every cycle, the clusters stay in the same location. He can do the base calling. So when you just follow this one, this cluster, you can see that in the first cycle, a T was called, in the second cycle a G was called, in the third cycle a C, and in the fourth cycle a T. And in this way you build up your reads, the sequences of every cluster on your flow cell. So this cluster will correspond to this sequence. So the sequences are called reads. Now, I always say you here, but it's of course the software from Illumina who is doing this, and you don't have to do this. So in theory, it is possible to go up to 300 sequencing cycles, but in reality, that's never done. Yeah, they typically stop after 100 cycles. So 100 cycles, that means that you will get reads of about 100 nucleotides. So the number of bases in your reads, or the length of your reads, is equal to the number of cycles that you do. And that's because in every sequencing cycle you build in one and only one base. So you can check for overlapping clusters. How? Well, because if you get overlapping clusters, then in many cycles they will emit a different signal. One will emit an A and the other one will emit a C, for instance. So you will get mixed signals when you have overlapping clusters. And the software can see that. He gets something that looks a bit like an A, but it can also be a C. So he's confused. And this is why the software not only calls the bases, but it also calls what is called quality scores. So for every base call that he does, he will also give you a quality score, how sure he is about that call. And that quality can be very high. It means it was clearly a G because the whole cluster was emitting a G, or it can be a low score, and then it can be more like, yeah, I saw a lot of signal there, A's and C's, so I don't know what to call. So if you have overlapping clusters, your base calls will have low quality scores, but we will come back on that.